Good morning or good afternoon or whatever time of day it is for you. Welcome to our quarterly safety working group scientific webinar. Today, we will be talking about the task force on interactive safety graphics will be highlighted. My name is Susan Mayo. I'm a senior mathematical statistician at FDA. And I and my co-host of this webinar, Michelle Zhang, Vice President of AFAMED Therapeutics, are happy to bring you this webinar today. Just a little background, the Interactive Safety Graphics Group is a task force within the broader ASA Biopharm Safety Working Group that's developing graphical tools for the drug safety community. The group is comprised of volunteers from the pharmaceutical industry, regulatory agencies, and academia to develop creative and innovative interactive graphical tools following the open source paradigm. A variety of tools for adverse events and lab analyses have already been released as part of the Safety Explorer suite and the Hepatic Explorer. The ISG group has continued to innovate, and 3 new interactive visualization tools are the subject of our webinar today. Now, if you have been a study of graphics, you'll know that the human brain is a particularly good has a particularly good pattern recognition tool, in fact, even better than computers. And so in terms of identifying signals, if we can use graphics design graphs well, we can actually use that pattern recognition capability of our brains to best effect in finding signals. So without further ado, I would like to turn it over first to Jim Buchanan, President of Covalence. Jim has been involved in this task force since its inception. We also today have Spencer Childress from Gilead Sciences, Preston Burns from Sarepta Therapeutics, and Natalia Andriachik from Pfizer, with a discussion from Cynthia McShay from UCB Biosciences. Over to you, Jim. And again, a reminder, please, if you haven't muted your phone, please do so.
Thank you so much, Susan. As Susan had just mentioned, the ISG Interactive Safety Graphics Task Force lives within the broader ASA Biopharmaceutical Safety Working Group. And in fact, that group is comprised of 3 work streams. The first one, interdisciplinary safety evaluation, is the home of the Interactive Safety Graphics Task Force. And I'd like to give a shout out to the members of that task force who, as she also mentioned, are completely all voluntary. So it's really people stepping up and volunteering their own time and hard work that has made this entire thing possible. For those interested, down at the bottom is a link to our webpage that describes it a little bit further. We had a number of guiding principles since the inception of the task force. First and foremost, we wanted these tools to be open source, that is free to all interested parties that wish to use the tools. But importantly, also, it means that it's transparent. The code is available. You can see exactly what we're doing, and it's customizable. So you could even take the code, bring it into your own shop, tweak it to your heart's content, and make it suitable for any particular use that you might have. It's been a highly collaborative path. We've engaged with data scientists and programmers and statisticians and clinicians in bringing together all the thoughts and skills of these folks to come up with these tools. And we think it also is going to foster collaboration amongst users, safety scientists, getting in touch with their biostats people and working together. So we really enjoy the fact that this is a highly collaborative effort. Certainly it's interactive. That's in the title of the task force. And that's been very important. We want people to be able to readily explore their data. It's definitely easy to use. It appears in a simple web page. And in fact, where it's even more important and the tools may be a little bit more complex, we have a clinical workflow that also helps to make this easy to use. It's data standard compliant. We can import CDISC data in Atom or STDN by default. But importantly, it doesn't have to be necessarily in those data formats. It could be just simple spreadsheets. We do a little data mapping. So we're quite flexible in the way that we can use data. It also is an agile system. We make frequent releases up on GitHub. And it's engaging in the sense that there's a path for feedback. In fact, we really enjoy hearing back from users because it helps us understand what additional functionality we should be adding and considering in future releases. So we start with looking at various types of study data that one might use. In fact, we can pull information from a number of data domains, adverse events, labs, demographics, and so forth. And then together with the chart specifications can result in the production of a variety of different views, if you will, of the data. And that's been one of the key features of the Safety Explorer suite. For those that are interested in a little bit more detail, there's this paper that we published some time ago that describes the Safety Explorer suite in a bit more detail. And then one of the most interesting, I think, new interactive tools that we had developed was the Hepatic Explorer. This was taking the original FDA's eDish graph and turning it into an interactive type of tool. Not only do you have the capability of observing all the data, but then also drilling down to subject level details. But this is also accompanied by what we call the clinical workflow, recognizing that not everyone are hepatologists by any stretch of the imagination. The clinical workflow allows the non-hepatologist to be able to go through a series of explorations, all accompanied by a textual description of what's the meaning of the test that you just performed. And it's all referenced back to the primary medical literature. There's also an article that we published if you're interested in a little bit more detail on this particular tool. But of course, you can always download it and work with that as well, too. These tools have been used, we know, out in the real world. And in fact, this was an article that was published some time ago about the use of these tools for a data monitoring committee. And so it's nice to see some examples of how one can actually use this in the real world. And just by way of anecdotally, in fact, just last week, I got an email out of the blue from a physician who sits on a DMC and was basically saying that he really enjoyed how it much simplified the task of going through all the lab data and so forth. He was very, very appreciative of the tool. And it's another example of some real life experience that this is something actually that people find of some value. So with that, I'm going to stop sharing and pass this on to a discussion of the volcano plot.
Thanks, Jim. All right. All right, I will share my screen. Oh, I think the host has screen control right now.
Just thinking about how we react to speakers who share their experience shows this inequity. I can't see an view, but I can imagine.
If everybody could mute themselves, that would be super.
Problematic substance use.
Yes, Michael Weiss, could you mute yourself, please?
Or give me like a little.
Spencer, you probably can mute him from your end.
And while I introduce myself as hi, my name is Steven, and I'm a person who uses drugs.
Okay. I think I need host control.
Yes.
Yeah.
not that I want applause for being an overdose survivor, but also think about how that impacts other folks who are in the room who may be afraid to share their living experience, knowing that we live in this abstinence.
You can. Okay, let me mute him and then pass it to you. Is that okay?
Oh, great, thanks.
Software esteemed channel.
Spencer, you're on mute.
Thanks. And I think I just need
Yes, I will.
yeah, host control. Oh, great. Thank you. Okay. All right, so let me pull up my slides. Okay, so I'll be talking about the Volcano plot, which is an app, a Shiny app that the team has developed, and its purpose is adverse event incidence analysis. And so I'll be getting into the background of this approach to incidence analysis, and we'll take a deep dive into how it's visualized, and then we'll have a little demo and just go over a few improvements that the app could use. So let's get into it. So adverse event incidence analysis helps describe an intervention safety profile. And if you have any background in statistical programming or clinical trial submissions, you might have seen one of these adverse event tables. It's presented by system organ class and preferred term, and you just kind of have the count of participants who experienced the event in each treatment arm. And these can get super long. They're super verbose, and so data visualization is a good solution to that problem here. This sort of data is also captured in something like a combination dot plot and relative risk plot. So this is a nice condensed view of the same data. You have the preferred term or system organ class over here on the left. In this left pane, you have the risk rate, or the incidence rate across the treatment groups. So you can see one treatment group's captured in these triangles and one in these circles. You have the percentage of patients experiencing that event. And then on the right, you have the relative risk of experiencing the event between the two groups, and you can kind of see, okay, so these events are more frequent in this group. So this is a nice plot, and we'll come back to it, but this can also get long because you can have just a ton of different events occurring in your trial. So as kind of another approach, the Volcano plot was developed. And you can see it saves a ton of space. It's captured in the dimensions of a chart, so you can just drop it in to your report, or it's just really easy to ingest. It emphasizes the safety findings, so everything above this red line is potentially significant, worth looking into, and it minimizes noise. So all of the adverse events that kind of aren't significant are kind of overplotted down here at the bottom, and you can just kind of ignore them. And then you can also incorporate multiplicity adjustments, which is probably appropriate for a more rigorous statistical approach here. But yeah, it's a great plot and very concise. So let's get into the aesthetics of this plot. So on the horizontal axis, you're plotting your statistic of interest. That might be the relative risk ratio or the difference in proportions between the two treatment groups. And then on the vertical axis, you're plotting significance, and it's a negative log scale. So as significance increases, the point moves up, and it kind of separates itself from the rest of the points.
小蛋蛋，你
And then,
OK 吗？
And then points are sized by the frequency of that event. So just mapping, the number of events to the area of the point. And finally, the points are colored and saturated by the significance and which direction the risk runs. So in your intervention, in this example, red points are more prevalent in the interventional group, and then blue points are more prevalent in the placebo group. So that's kind of like the explanation of the visualization in this chart. So I think now we can get into a demo. So I've got it pulled up over here. And let me take a step back. So this is the safety graphics framework, and this is kind of a user interface that lets you dig into various safety domains like adverse events and labs. This might look familiar if you've ever played with the app, but you can map your data, your columns. You can filter, set an analysis population. I don't want to steal Natalia's thunder, but you can look into individual participants, and then you've got this list of charts that kind of pertain to various data domains. So we're gonna look at the volcano plot, and this should look pretty familiar. Right now we're plotting risk ratio. That's the x-axis. Y-axis is negative log of your p-value, so higher points are more significant in the comparison. We're comparing in this kind of example data, we're comparing this interventional treatment group to placebo, I think. So these three events are going to be kind of potentially significantly more prevalent in the interventional group. After you click on a point, you can scroll down and look at the comparison. So the p-value here is 0.0006, and here's the estimate is the risk ratio. We're comparing the reference group is placebo, and we're comparing our interventional group to placebo, and we're looking at risk ratio. We can also look at the absolute risk difference, and here, again, this is just subtracting the prevalence in one group from the prevalence in another. So we're looking at this by system organ class, preferred system organ class. We can also look at it by preferred term, which will turn up more points because it's a kind of a subcategorization of system organ class. So we can get a little more granular and look at more specific adverse events. And finally, I just added this, but we don't have a multiplicity adjustment. But if you wanted to, flip or adjust your p-value, you can do it right here, and it'll kind of change which events are considered significant. Yeah, so that's the volcano plot, and let me flip back to the slides. So some improvements here. We could add a multiplicity adjustment, but again, this is a tool for exploration, so it's maybe not appropriate in that scenario. But we could add something simple like a p-value slider, which that dropdown kind of addresses. But other multiplicity adjustments are Bonferroni, or what's referenced in the paper that kind of debuted this volcano plot as the false discovery rate. And then another improvement we could make is to incorporate time intervals to look at specific time spans in the study. So maybe the first week or two after treatment start, or after treatment end, to kind of really hone in on specific spans in the trial. And then, as you can see, we've already incorporated this in safety graphics, but it's kind of like a custom configuration. So it'd be nice to just include it by default, which would be as simple as adding, updating the configuration of the safety graphics library. And then finally, we could really stand to improve some aesthetics here, get some column headers on these tables, some, more descriptive column headers, and just kind of make it a little prettier. But I think that covers it. So I'll stop sharing and pass it over to Natalia. Preston.
Thanks, Spencer.
All right. Do I need to give up host control? Oh, there we go. Thanks.
Okay, can you see my screen?
Yep.
Okay, perfect. Okay, so I'm going to talk about the NEP Explorer. So this is a renal safety monitoring focused R package for acute kidney injury evaluation within clinical trials. So we use the KDIGO and delta creatinine systems for classifying changes in serum creatinine, placing them into stages. There's an R-Shiny application included within the package that provides some interactive pieces, so you can hover and drill down, change settings, zoom in. And all of that is to facilitate exploring kidney function, both across the population of the study and within a patient over time. Along with this, Jim sort of alluded to this earlier, is there's a clinical workflow that's associated with it that'll provide guidance for users on how to use it and what are the key assumptions of the tool. So the primary visualization in the NEP Explorer package is this creatinine scatter plot. So in this scatter plot, each point corresponds to a patient, and the location of the point in the scatter plot is based on the maximum change in their serum creatinine, both the absolute change on your y-axis there and the fold change on the x-axis there. So as you can see, a point in the red area has had a very large change in serum creatinine from baseline. And according to KDIGO, fold change criteria, they would be in stage 3 if they're in red. And within the chart, there is a bunch of functionality for zooming and hovering and as well as clicking on patients. Along with the scatter plot, we have just a summary table of how many patients and what percentage of patients fall into each stage. So if you look at this table, we would say, okay, there's 3 people who fall into the stage 3 classification based on their fold change in serum creatinine, and that's 3% of the study population. And if you click on a point in the scatter plot, and I'll show you this in a demo in a moment, you get a longitudinal renal profile for that patient. So key renal markers, electrolytes are all sort of displayed over the course of the study for the patient, so you can explore a little deeper. And in the upcoming year, our goal is to get this more production ready. So we want to integrate with the safety graphics framework. So just like Spencer was showing, in that safety graphics dropdown, how you can choose volcano plot. We want to get NEP Explorer there so you can use it within safety graphics. We want to integrate it with safety profile, which is a really cool package that Natalia is going to talk about in a moment. We want to have real world pilots, develop more of our documentation, finalize that clinical workflow, as well as get feedback from academic industry and FDA nephrologists. For example, we met with Dr. Sarah Jandeska earlier, or I guess last year, and she provided some fantastic feedback as a pediatric nephrologist, kind of around, what age range would this work for or not work for, and things like that. So we're looking to get more of that feedback. So with that, I will jump into demo and show you the application. Can you see my web browser?
Yep.
Perfect. Thanks, Natalia. Okay, so as you can see, this should look familiar. We've got our scatter plot here, and if we hover over a point, let's choose this one here. You can see information displays for that patient. Okay, this is 04029. This is classified as a stage 3 KDIGO change. That there's a 5-fold change for them in their creatinine. You can see their baseline there. Milligrams per deciliter is 0.28. You can see at the bottom what study day, what visit did this change occur on? Would this maximum change occur on? It happened on study day 61, and it happened at an unscheduled visit after their first visit. And so you can hover around in here to look at information for patients. When they get clustered together and it's hard to see, you can zoom in on the plot. And that kind of allows you to access everyone's information that you want to get to. And then we've got this table over here that I talked about earlier, where this is sort of your study level, safety signal. Okay, is there a kidney related safety profile here? Just at a high level. And then in this plot, you can actually click on the patient. So if I click on that person we were just talking about, it will pull up these profiles. So at the top, you've got demographic information, what's their age, sex, race, what arm are they on in the trial. And then down here, we have over time, their creatinine and cystatin C values, both the percent change and the raw changes. You can see their EGFR over time, their electrolytes, the fold change in different electrolytes over time, so phosphorus, potassium, sodium, so on, their blood pressure, and their their normalized albumin over time as well. So the last sort of feature I want to show you that's sort of newer is we also added a time animation. So if you turn this on, you can either by study day or by visit view specific points in time, or sort of play the course of the study. So if I choose visit here, you'll see here's our little axis. If I hit play, the chart will go over the course of the study. So interactively show what were the changes at each point in time. And then you can drag this and just look at a specific piece of time. So if you want, like I'm interested in visit 6, I can drag there. Okay, this is how things were at visit 6. You can change the speed up here. So if I drop this down, maybe, and this is sort of the reason the speed is relevant is, based on the number of visits or days you have, you may not want to, that determines how fast you want this thing to move. But if I turn it down a little bit, oh, just kidding, I turned it up. That's on me. So if I slow this down, you can see here, I have a little more time to see each visit. And I just want to acknowledge, there was a whole team of developers and clinicians and people involved in helping produce this. And I want to hand it off to Jim to talk about the clinical workflow associated with this.
Great. Thank you so much, Preston. Yeah, so not to get into the details of it, but just to let folks be aware that once this is released, it also will be accompanied by a clinical workflow similar to that which we had developed for the Hepatic Explorer. What this will do is it's part user manual, give you some understanding of the functionality and such, as was just described. It also goes into some discussion of mechanisms of acute kidney injury. So one can get a sense of pathways by which this would appear in patients, particularly as it relates to drug-induced kidney injury. And then, similar to the Hepatic Explorer, it'll go into a diagrammatical display of a number of sorts of analyses that would be recommended to perform, which one can do using the information that's presented in this tool. And so, for example, the extent to which you have cystatin C values available, that's also available to be utilized in the evaluation. But if not, there's still other analyses that you could do. The goal is to be able to identify instances of acute kidney injury. Now, we've taken a little bit of liberty with these two criteria classification systems, KDIGO and delta creatinine, are really intended to look for changes within a week apart. And that's just not practical in most clinical trials. We're not getting labs every week throughout the trial. So we've taken some liberties, if you will, with the application of this. But nevertheless, it's still something that is nevertheless useful to be able to look at the totality of the data and to be able to pick up, especially with that animation, when these aberrations might occur at various points in time, not just in the very beginning, which probably is where the risk is greatest, but also the possibility of picking this up later on in time. Definitely there's some caveats right now. As Sarah had pointed out, this is not intended for a pediatric population. This is assuming it's an adult population that is otherwise generally healthy without chronic kidney disease and so forth. So there's a few caveats in the type of patient population that this would be applied to. But nevertheless, at the end of the day, we hope this will be an easy way to be able to pick up instances of acute kidney injury, and then also with some of the exploration of the data, start to get a sense of what might mechanistically be underlying this. Okay, folks, thanks. I will turn this back to, I believe, Natalia.
Yep, thanks, Jim. I just need the presenters view, I think. I'll just wait for that. Perfect. I think I have it now. So let me share my screen. Okay, and hopefully you are able to see it. So hello, everyone. My name is Natalia Andrichuk. I'm a data scientist at Pfizer, and I'm also part of the ISG group, as you probably have noticed already, and one of the developers of the safety profile. And today I'd like to give you the overview of the safety profile. But first, I would like to introduce the development team that worked on the safety profile. Those are the people who contribute to the code or to the design of the tool. And we're a great team that, creating the safety profile. And this is the newest renderer of the safety graphics universe. And we're on track for the version 1.0 release later on, but it is in a good shape to demo it for you today. And we're also working on deploying the tool to the Shiny Apps IO platform so that anyone can go and play with it without installing and get a feel for it. And let's discuss what is the safety profile. So the safety profile package contains a Shiny application that allows users to view subject-level profile reports for patients that participate in clinical trial. So visualizations and listing included in the package, they present a patient-centric summary for different parameter changes over the trial period. It includes the laboratory results, the concomitant medications, the adverse events, and participant demographic data. The safety profile itself can be used as a standalone Shiny application, and this is what I'm going to demo it for you today. And at the same time, it developed with the Shiny modules that allow the tool to be a part of the safety graphics R package. So Spencer gave us a sneak peek when he showed us the safety graphics demo, and it had like a profile icon on it with the safety profile embedded. So this is what I'm talking about. This is a more robust safety monitoring framework. And while there are some other libraries that will give you the safety profile features and charts, the one that we developed just works and integrates seamlessly with the safety graphics if you're already using it. The package is not yet on CRAN. So you can install it from GitHub if you're eager to start playing with it and look at it. You can do the DevTools install GitHub, and we ask you to reference the main branch because it's the most comprehensive one. And then you can just run standalone app. And that's that's it. Let's go into the demo and just take a look at the safety profile together. Okay, so this is the standalone application. This is the safety profile. And when you first open it up,
could I have one slice of the capital?
and if you could
Yeah.
mute yourself,
And,
that would greatly help me because it's a little bit loud. Thank you. So when you first open the safety profile, this is what you see. So on the top ribbon, you see the participant details. So there's a participant dropdown, and you can change the participant, and the data and the tabs will change accordingly. And then you also see some additional demographics details for the participant, like the treatment, the sex, the race, and the age columns. And the overview tab that you will see upon the opening the app presents you with the participant data listings. And you can select from preloaded domains. So domains I have available for now are labs, demographics, adverse events, and concomitant medications. And below that, you would see the listing. And if your listing is pretty wide, like I have right now, you can click on the row, and this will help you when you scroll to the side of the listing. And the table, the listing itself, is also searchable. So let's say you are looking for only albumin details, so you can search for albumin, and only the relevant information will appear in the listing. So this is pretty much it. This is just the listing explorer, the overview. Let's move on to the next tab, which is event timeline. Let me pick my favorite participant. So the tab consists of the chart on top and the listing below. So the chart called study event timeline, and you can see the subheader. It displays the number of events you see in the chart. They just broken by events. So the chart itself has the x-axis for the study day, start and end day, and then the y-axis shows you the events. So depending on the start and end day availability in the data, the chart would either display the event as a dot if there's only one date available, or as a line if there is a start and end date available. And then you can also hover over and see the details for the given event in the tooltip. And the legend will show you the domains. And then you can see also there is a note here, how many rows, if any, were dropped because from the chart because they had missing start and end dates, because it's impossible to visualize something that doesn't exist. However, you find this information in the listing below. So the listing below will show you the events that are displayed in the chart, and also the events that didn't have the missing start and end dates. And the listing is also searchable. So let's say you only want to see the concomitant medication part, then you will search for CM, or maybe you're only interested in the severity for the mild events. So you can also search for that, and listing will filter the appropriate data for you for a given participant. And it doesn't matter which tab you're on. You can you can always change the participant, and the data will be changed accordingly. So this is pretty much it for the event timeline tab. Let's move on to the measures over time. So the measures over time tab includes the dynamic listing for labs available for the selected participant in the dropdown. So at a glance, you can see the measures, the upper and lower limits of normal, and then the trend for this particular participant visualized as a spark line that also has the hover over details. You can always expand the details, clicking on this little plus sign. And then once you expand, you'll see the additional details. For example, visits and study days and the results for each visit and study day for this particular measure for this particular participant. And again, this listing is searchable. So if you're only looking for calcium, you can start typing calcium, for example, and the search bar will match the appropriate measures for you to view. And this concludes the demo of the safety profile tool. And as I mentioned, we're still tidying a few things up for version 1.0 release, but it shouldn't stop you for going in and playing with the tool. So thank you so much. And I'll pass it back over to Jim.
Great, thank you so much, Natalia. I have to say, the patient profile is a functionality that we've been working toward for quite a while. It certainly became apparent in the drill down capabilities for a lot of these tools that to be able to more fully see other useful pieces of information would greatly help in the evaluation exercise. So, being able to match up maybe the onset of a particular lab excursion with an exposure to a ConMed, or were there associated adverse events with that laboratory abnormality would be very helpful. So we're really looking forward to not only having this perhaps as a standalone, but also being able to offer it as part of the drill down capability for these various tools. For example, you're in the Hepatic Explorer, you see some points up in the potential Hy's Law range. We already have the capability of drilling down and looking to see how various analytes are moving, but to also be able to drill down and look to see the pattern of adverse events and ConMeds and so forth would be extremely useful. So we're definitely looking forward to having this capability accompany a number of these tools. Natalia, I'm sorry, or maybe Susan, I will pass the show back over to you.
Thanks, Jim. We're running a little ahead of schedule. So I noticed one thing in the chat. Someone asked if you could demo how you read the data in. Natalia or Spencer or Preston, would you be willing to show that, please?
Yeah, I can maybe do a quick little.
Thank you, Spencer. I know it's on the spot.
No worries. I've got R pulled up. Do y'all mind sharing the host capacity? Thank you. Okay, I'll just pull up an R session real quick. So if you have your data locally, or if you read it in from your file system, network system at your company, you can pass it directly into the app. So for instance, here, let me make this a little bigger. So on the left-hand side, you can see we're calling Safety Graphics app, which includes another library called Safety Charts. That's all the charts in the dropdown we saw in the app. We have Safety Profile, what Natalia just demoed, and then this last load all is the volcano plot. So here, when we call the app, we can say something like domain data equals list. Then we can specify our demographics data set. And this is just example data we have in another repository, but you can see how you just pass in your own study data. But I'm gonna just reference our example data real quick. And that's basically how you specify it. It's a named list, DM corresponding to your subject level data set, labs, your labs data, AE is your AE data set. And assuming your column names match the expected column names in the app. This should kind of pull up for you pretty easily. If they don't match exactly, you can come over to this mapping tab and kind of, you know, choose the appropriate column name in your data set that maps to, for instance, adverse event start day. There are also value mappings on this view, so you can specify the treatments you want to compare in the modules that are looking at, you know, kind of comparing two treatments. And that's about it. So for instance, for the Hepatic Explorer, you can specify the analytes that go into that. So these dropdowns should populate with your, you know, with your lab measures in your data set. You can kind of like click through here, specify all of your column names, and then your app should run. And this framework will let you know, all these little green checks, it'll let you know when your mapping meets the requirements of each module. I think that about covers it. Trying to think of
Spencer, one question. I haven't used this for a couple of years now, but I seem to recall there's also a way after you get into the app to pull up a data set, which might be the way a safety clinician would be more likely to do it.
Oh yeah, to like kind of load your data interactively?
Yes, yes.
Yes. Yeah, I think that's here, let me back out of here. So alternatively, instead of, you know, kind of writing out code to point to your data sets, I think you can safety or init. I have to go look at the code real quick. So yes, there is a user interface if you want to load your data interactively, and I think you call safety graphics init. I haven't actually run this in a while, so I'm not sure it'll run, but it's a little buggy.
Okay. Sorry.
But
Again, I
yeah,
didn't mean to put you on the spot, but I guess when you release this, or there probably is some training for this.
yeah, there should be a vignette if there's a
Yeah, Jim, you've used this before, right, as a clinician?
Yeah, exactly. And then I am not a programmer, but I just, you know, have my little couple lines of code that I copy and paste into RStudio, you know, and it runs. And, you know, Spencer just demonstrated, when you finally get to the point, the safety graphics init line, then it will display that web page, and you haven't loaded data yet at that point. And so it offers you to load your data, which I typically have, you know, as a separate external file. And it's just like, you know, uploading data in any other program where you're looking for a file. You just navigate to the folder where your data set lives, click on that, it loads it. And then I typically am getting lab data that's not CDIS compliant. You know, it's just come straight out of the lab. And so they're calling the headers by any of a number of names. And so invariably I got to go in and say, okay, where's the ALT? Where's the lower limit of normal? You know, do that mapping function, which is very straightforward because it's just a drop down menu that you pick from. And then you're good to go. And then you've got your data loaded, you've got your graphics then available to choose from. And it's pretty straightforward.
Thank you, Jim, and thank you especially, Spencer, for demoing that on the spot. And I learned a little bit more about what's behind the tool. Thanks. So now we have time for Cynthia McShay from UCB. She has some discussion points to make. Cynthia, I believe you've been using this some in your organization.
Yes, Susan. And for those on the call, my name's Cindy McShay, and I am a safety statistician. I lead a team of safety statisticians at UCB, and we have been using on a small scale the application more as individuals. We are exploring how we can deploy it or use it for broader use in our company. When I say we, I'm talking about members of my team and also people who are on the work stream that have been on my team. One who, Lovemore, a shout out to you, is on the call today, but that have helped or helping with the deployment. So I'm here as a discussant, really as an end user of the ISG tool, not as a developer or programmer, but someone who does exactly what Spencer just did. Go and take the tool. Either I've used it directly from the CRAN or from GitHub, but also now use a version that's had some adaptations really for that data load piece, the interface to streamline that sits on one of our UCB servers. So my use of the tool is for data exploration, to do an informal look at trends, outliers. Love the drill down capability, make a lot of use of that Hepatic Explorer, plus the AE, some of the existing tools, the AE Explorer, the timeline plot. I'm really excited about what was presented today and how we can use that. But I will say the use is strictly for data exploration. It does not replace our formal analysis or our proper analysis that is governed by our statistical analysis plans, where we have what we refer to as a GXP compliant deliverable. So we use it as a supplement, really to look at the data without having to, in an interactive way, without having to actually write code ourselves to do it. We also use, or I'll say I specifically use, the HTML download function so that I can help other people on the teams I work on look and explore the data, or I use screen sharing, just depending on the scenario, to share the interactive graphics on the screen. And so, but in every case, it's for non-GXP use. And we are looking, it is deployed on a server, but with limited use right now to mainly to safety statisticians and in a non-GXP capacity. We are looking into or exploring the version that we have with adaptations being validated for GXP use, but that's still very much a work in progress. So, so I'm happy to answer any questions about our use, or any questions you have about how you'd use it in your day-to-day activities. I really advocate the use of the tool. We've had great, I've had great success using it. And so at least in an exploratory way. So I know you probably have questions for the developers of these new tools, which again look really exciting. And I'm happy to address any questions that you might have about my use. Thanks. I'll turn it over to questions. Any questions from the group? Or I see there's questions in the chat.
Yeah, there's questions in the chat, which we can get to in a moment. Does anyone have a question for Cindy right now? Otherwise, we can start at the top of the chat. Thank you, Cindy. Thank you for sharing your experience. Looking at the top of the chat, I see Wei Wang has a question about the volcano plot, and Spencer, you addressed that. Would you like to say anything more about that, Spencer?
Yeah, I think Wei was mentioning the original proof of concept volcano plot has a nice color legend that's makes it a little more interpretable, which adverse events are more prevalent in which treatment groups, and that's definitely something we could use in the app. So yeah, I filed an issue, and I think it's a great idea.
Thank you, Spencer. Eliza Thompson, who is a specialist in the area of kidney injury, who Jim and I have had some discussion with, mentioned that something about AKI. Eliza, are you still on the call? Or you may have needed to drop off. Okay, so she says that it may not be AKI. Jim, would you like to take this one, or maybe Meng Chun?
Yeah, certainly. She's correct in that there are some nuances in terms of the wording that we choose to use when we're talking about damage to the kidney, and so she's correct in that we need to be careful in how we are describing these sorts of injuries and such. So we'll certainly be going into more detail about that and the differences between injury and damage and so forth in the clinical workflow. So I was probably maybe a little too cavalier in the use of the term.
Okay. Okay. So that didn't have to do with the plot so much as maybe some comment you made.
Yeah, it speaks to how we want to define damage to the functioning of the kidney in several sort of parameters. I mean, one of it has to do with the time to onset for these changes, the time period in which we're looking for evidence of these changes and so forth. So it definitely gets into more of the detail of how one goes about looking for these sorts of changes to kidney function. But rather than going into that here, it'll certainly be explained in a lot more detail in the clinical workflow. So just to give people sort of a heads up that there are some nuances that you need to be aware of.
Yeah, and this might be a good time to say that with an interactive tool, one thing we realized is that you can kind of get lost in there. And so it's important to have a workflow so that not only do you have a tool that's set up that allows you to look at various things, but also you know specifically what the tool is designed, to do first this step and then that step and then explore this, etc.
Yeah.
Which is different than a static graph.
Well, and I think the other thing that would be useful in being able to provide the clinical workflow and some description about nephrotoxicity and so forth is to hopefully maybe get everybody using the language more consistently. So that when the safety people are talking to the clinicians, when they're talking to the regulators, when they're talking to principal investigators and so forth, that we understand the choice of the words that we're using that mean specific sorts of things. And so hopefully there'll be a little bit more continuity across stakeholders.
Okay. Okay. Next I see Mike Hollopy, you had a comment about efficiency of the programming with large data sets. Did you have anything more you wanted to add there? And then Spencer had a response as well.
Yeah, it was. Oh my God.
Okay.
Oh, okay. I'm back. It was just I know that when Jeremy programmed the Hepatic Explorer, that was several years ago. And probably if he were to reprogram it today, there's probably newer packages, could probably handle the data in much more efficient ways. And I was just wondering if the Renal Explorer incorporated any new programming packages that would make the efficiency for handling larger data sets possible.
Yeah, I can speak to that a little bit. Yeah, so Renal Explorer, so Spencer pointed out we're using R now for things. I guess maybe you could like think of there's maybe two sources of slowdown. Like one would be if you're doing any math, and you're doing that in the browser, and then the second would be actually plotting on the page. So like if you put 1,000 circles on the page, you know, your browser slows down. So that's just, I guess, context. I don't know. I think we'd have to test it. I don't think we've tested it with a large data set. So I think that's feedback for when we do our real world pilots. We want to make sure we try it out with a lot of data. I mean, at some point, right, like if you have 1 million points on a scatter plot, you can no longer read the scatter plot. So there's like an upper limit in terms of interpretation. But you're right. I think that's something we need to test. I think, like Spencer said, I think it would be more performant, but I think we should test it because there is JavaScript used, you know, by R to plot these things because we're adding that hover functionality and stuff. And it is, I think, depending on how that's implemented, it could still be slow if there's a ton of stuff.
Yeah, in the Hep Explorer, all the math is in the browser. So you're loading all the data and you're doing all the calculations. And I think in the Hep Explorer, it's
an R. Yeah.
It's an R.
Thank you.
Yeah, when we have folks participated in the beta testing, that might be one of the useful things to test is how large a data set can you load and still see that the tool function.
Yeah.
Yeah. Someone's keeping a log of testing for beta testing.
I'm writing it down right now.
Thank you, Preston. Someone asked, can you get the slide deck? Can we get the slide deck and the web links? Michelle, would you like to address that question?
Yes, we will share the slides after the webinar.
Okay, and how will those be shared? Because I think people found this webinar in different ways. Will that be on LinkedIn?
Probably on our website.
Okay. Okay. Can you share a link to the website, please? So people can look.
Okay, sure. Yes, I will type it in.
Thank you. Thank you.
Okay.
Okay, and we already talked about reading the data. Ranjita asks, is there a way to flag extreme low and high values in the measures over time tab? And how about in aggregate across all subjects and all analytes? And Natalia addresses that. Ranjita, would you like to ask more? Natalia, would you like to say anything further?
It would be interesting to see because I think it's helpful even from ongoing data review and cleanup process for an ongoing study as well, particularly when you get local labs, and I'm assuming local labs are also mapped into the lab tab. You know, you're not waiting till the end of the study to clean up some of these errors that might have been just maybe sometimes purely because of the translation of units.
Yeah, I think that's a great idea, and we can color this out, I think, in measures over time when you look at the individual participant, and I highly recommend to check out the Safety Outlier Explorer for that matter, when you can see the outliers right away across all the participants, and it's a well-tested tool already within the safety graphics, so you might find a great use at it.
If we have after all the questions, if you wouldn't mind just showing that real quick, demoing will be really great.
Sure.
Thank you.
Thank you. And Ranjita, you had another question about cystatin C.
Yeah, that was just kind of out of curiosity, just how many folks actually see cystatin C, which is kind of not really used clinically. It's more of a, exploratory biomarker of renal injury. How many of them are actually using it to get a sense of how accepting are the agencies, not just the FDA, but outside of US as well, in the monitoring of that analyte.
Yeah, I have to confess, it's not typically captured, it's not part of a standard panel. Not unless you're actually doing a study where you expect to see some renal effects, then in those circumstances, I've seen the development team add cystatin C to the lab data being collected, in which case we can't plot it. But as Spencer points out, if it's not in the data set, it's just something to plot. That being said, I had a client recently where they were seeing acute increases in serum creatinine after the start of study drug. And it started to look like it might have been a creatinine transporter inhibition. And it would have been exceedingly helpful at that point in time to have cystatin C, because then you could have quickly determined that, in fact, it's not a nephrotoxic effect, it's a transporter effect, because the cystatin C wouldn't be moving. But unfortunately, they didn't have that in the data set. So, yeah, maybe at some point where there starts to be, renal signals, people will then perhaps incorporate cystatin C in subsequent trials in order to nail down the mechanism. But you're absolutely right. Right now, it's not commonly encountered.
Got it. Thank you.
Thanks. Then Gary Rosner has a question about, does the safety profile show or allow for showing the different grades on the same adverse event over time, and possibly by color? And Gary, would you like to add any more to your question? Otherwise, we can go to Natalia with a response. And Sharon Murray also suggests a star or other marker for SAEs would be good. So, Spencer?
I can just chime in. We don't have that implemented on the safety profile tab because we're kind of aggregating across data domains, adverse events, concomitant medications, exposure. So those are kind of occupying that color aesthetic, but in the adverse event timelines, which is one of the chart modules in the dropdown of the safety graphics framework, that's dedicated to just adverse events, it's colored by severity, and it also has a serious adverse event highlight built in, so you can kind of get at that information in another module.
Very good.
And I can add to that, Spencer, that if there are typically when data are reported in adverse events, or at least I know conventions at our company, is if there's an increase in severity of the same event, that it'll be recorded as a new AE with the higher severity. So you can see the progression in the module that Spencer just mentioned. You can see basically the span of the AE activity for that event over time for each patient, but it's a different module.
Very good. I love how much feedback we're getting on for the team on suggestions for other features, and also other modules that weren't highlighted today. Let's see, Mike Litzinger.
Yeah, Susan, it's like we were talking about in the beginning. The guiding principle was to be engaging.
That's right. Yeah, Mike, would you like to ask your question?
I think it's largely been addressed, but I did want to point out in the both for just line graphs as well as in the profiles for dose escalation studies, this is really helpful to be able to see this visually as a color or a shape in the Z dimension. I do realize this is a different trial structure than your typical placebo active type of thing, but if there's some way to incorporate that, it is much more complex. I will give you that. It could even be just putting it into the tip, or making sure that with your adverse events or your labs that there is in the tabular displays. But that's a functionality that I can give you a lot of feedback that medical monitors and scientists really are looking to know that. When they see an extreme creatinine value, for example, they want to see the attributed dose level.
Absolutely.
Thank you, Mike.
A lot of sense.
And
Yeah.
that's one of the most wonderful things about this team is that it's clinicians and data scientists working together.
And I guess I'll add to that, Mike, from the end user side, because I know that I can and definitely say understanding what dose people are on when we see these events happen. But that's we can use the other deliverables, at least folks that I've been working with that they have. So because you can drill down to the event and with the listing, you can see the dates. So it's manual. It's not as interactive. It'd be fabulous to have it in the interactive tool, but you can still bridge to the information you have via other presentations to get the dose levels.
Yeah, and again, that's very helpful. The key is the ease of this overall package as a URL that can be, accessed by a physician or a monitor or somebody that's just not very technically savvy.
Sure.
Perfect. Thank you so much for that, though.
I guess I'll throw one other thing in there is that the code, usually at least within safety graphics for the tools that are there, you can see the code. And so, this isn't for clinicians, but for statistical programmers or statisticians, the code can be borrowed. I think there's some of the interactive capabilities you have to disentangle with the regular code, but you can certainly use the code to produce a formal graphic, whether, and if you can use R for that with a validation. So it could be GXP to support communication to the team as well. And something they can use beyond exploration. And we've done that before and added the doses onto it. But it is more complicated and harder to standardize because the way dose can be set up in very different across different atoms, SDTMs.
Yeah, and I just
Thanks,
wanted
Cindy.
to point out that the safety profile will work with the exposure domain. So when you add it on to the list of the domains, when you first initiate the app, there is a way to add the exposure domain to it.
Okay, so I see there's another 11 messages here. So wanted to make sure we get to other questions.
Susan, there was one question I see about that was for me about does our safety team have access to this? I'm happy to field that. I don't want to jump ahead,
Sure.
cause us to miss anything. But the question there, that's a great question. And for safety graphics right now, not direct access. We hope, and we are in the process of exploring how we can put some governance around it so that it's clear what the use case is for the application. But right now the interface is together. They work with a statistician or it's used by our statistician teams only, but it's certainly in the plans to have it deployed. We do have a different application in-house that people use and it is deployed to our stakeholders, but it doesn't have a lot of the nice functionality that we see in safety graphics. So we're working on it, but we have not yet completed that.
Great, thank you, Cynthia. Appreciate it.
And then Connor asks if there's a way to load the data within the app, and Natalia kindly adds there's a vignette that talks about that in particular. I know that functionality is there because I've used it. Do any of the developers or anyone who's using the tool want to say anything more about loading? We talked about that already.
I just want to add on to, so this is the functionality that Spencer showed, and it did act a little buggy for him. I don't think we had many bugs reported regarding this, maybe because it's less used as the other functionality, the way we use the app when we specify domains, and the way Jim mentioned that you just copy this couple lines of code and you at the app. But if some of you use it and you definitely see something not working for you, please feel free to submit the issues for the GitHub so we can take a look at it and improve upon.
Thank you.
I think there was a comment too. I don't know if it was the same one about like doing it interactively within the UI, like changing the data set. So just bringing that up as like, I guess there's a deployment decision that happens when you're using safety graphics, whether you're going to have one app per study or one app for multiple studies. And if you've got one application for multiple studies, then you need to be able to choose studies within that app. But if you do that, it sort of goes outside of safety graphics. So you have to use safety graphics as a module at that point. And in front of it, in a larger application, you put a sort of a study or data selection step. That's just a
Okay, thanks, Preston. There's a question from Alan Wu about drug-induced liver injury, which I believe was the first module that we worked on as part of the interactive safety graphics. And Meng Chun mentions that's published. Natalia has offered the link to that, to get to that. Jim, do you want to say any more?
Yeah, that was the really first robustly interactive tool, the Hepatic Explorer. It certainly, you could use some of the Safety Explorer suite tools to identify excursions of transaminase and elevations of bilirubin and so forth. That could give you a sense of whether you might have some drug-induced liver effect. But the sole purpose of the Hepatic Explorer was to take that eDish graph and make it much more interactive, and then to support other ways of looking at the data, such as the NR modified Hy's law approach, for example. So, yeah, if one is seeking to find evidence of drug-induced liver injury, then the Hepatic Explorer really would be the principal tool I would recommend for doing that.
Okay, let's see. Pad Magic mentions it would be helpful to add exposure and medical history domains to the patient profile. And I noticed, Spencer, you picked up on that one. Would either of you like to say more on that?
Is this the volcano plot coloring by SOC?
This is adding exposure and medical history domains to the patient profile.
Oh, right. Yeah. Yeah, I think exposure, we have built that one into it. And I think we have a medical history timeline, or I think we can kind of include that one like we include adverse events and concomitant medication. So I think it's just a matter of creating, the mapping for that data domain, so it plugs into the profile and also into the safety graphics framework. So yeah, absolutely, we need to get medical history in there, and exposure should be in there by default.
Okay, Natalia says it's there. So great, thanks for the feedback, Pad Magic. And then the next one is about the volcano plot. Presenting by preferred terms and color coded by SOC, SMQ, FMQ, etc.
Yeah, I like this idea. So basically, instead of so when we're looking at the volcano plot by preferred term instead of system organ class, we can kind of color the points by system organ class, since that's a higher level term, or by, I'm not sure what SMQ, FMQ, EOI are.
SMQ,
standard.
yeah, go ahead, Cynthia.
I was just going to say standard medical query and FDA medical query.
Okay.
That's where you collect, it's a collection of preferred terms that kind of mean the same thing.
Okay, yeah. So any higher level term, we could definitely include that in a drop down so you can change the color aesthetic, and added that to an issue. So yeah, I think we can implement that one pretty easily.
Okay. Pad Magic, if you're still online, did you have anything more you wanted to say about that? I'm not sure what EOI is.
Events of interest.
Thank you, Jim.
Yeah, it's event of interest, and also like in the legend, you will see all this, the SOCs or the EOIs listed because they are color coded. PTs are color coded, right, with the SOCs. So you can see, all these SOCs in the legend. Suppose if it's color in the same way, like if it's color coded by FMQ, then you can see it.
Okay. Well, I've gotten to the bottom of the chat. Michelle, I'm ready to turn over to you unless there's any final questions. We do have a little bit of time yet. We have 7 minutes.
Susan, if I could just make a comment about the
please.
Yeah. So as folks noted in the display on the y-axis, there is a depiction of a threshold value for the calculated p-value. But I think we just need to make it clear that one needs to be very careful in utilizing p-value tests in the exploration safety data. We are not hypothesis testing. We're exploring the safety data. So we can't use the p-value to say, okay, this is a statistically significant difference, and therefore it has a particular meaning to it, because as we just said, there is no hypothesis testing. This is simply exploration. So we need to think about the p-value less as a statistical term that we're otherwise familiar with, but instead think about it just simply as a threshold above which there may be some degree of association. But other than that, we just have to be very careful that we can't apply our usual statistical terminology, if you will.
Right. And so adjusting for multiplicity is just it's not a hypothesis. So there's no need to adjust. It's can be seen as like a threshold value above which you're looking at those levels.
Exactly.
Mm-hmm. I totally agree.
This is Mary. Can I chime in a little bit on the p-value conversation?
Yes. Hi, Mary.
Sure. Hi. So I would even hesitate to try to use it with any kind of thresholds. It's what we try to say when we use it for safety, and we're big. Some of us actually like using it for safety, but it's just a number between 0 and 1. I guess if you take the log, it's different, but it just gives you an idea relative to your other events, right? How strong your evidence is for an imbalance. Because I think we would even still hesitate to tell our clinicians focus only on the ones that meet this threshold. I wouldn't even go that far, right? Because a lot of that depends on how severe the event is. What event are we even talking about, right? Some events you're going to have a different threshold in mind than others, and so it really is just a nice tool to give you an idea of how strong your evidence is for an imbalance. So I would hesitate, definitely what you said about not any kind of hypothesis testing, but I would still hesitate to even introduce the idea of a threshold. Because it really depends on the event.
So I hear what you're saying, but then how would you address that with respect to how you draw the plot? Because
the
you
plot
have to have kind of a level, above which you're making the circles.
I still think your most important ones are visually popping up, right? The top right ones, visually, those are going to be, the ones.
Okay,
I
see
think
what you're
that
saying. So
drawing
don't
a
worry
dotted
about.
line is controversial, right?
Okay.
You know, and not to say some of our users still wouldn't want to have a line just to give them an idea of where you are on the scale of 0 to 1 in p-value terms, given after you transform the data. But I still, even if that option is on a volcano plot to have a dotted line, you know, that's fine as long as people are, it doesn't go along with that. You should only pay attention to the ones who are above that line. Those are the kind of things we shouldn't say.
Exactly.
Right? You need to pay attention to everything.
Yeah.
But the dotted line can still help. It gives you visually maybe a little idea, but I would still hesitate to say, you know, focus your attention only on these, right? We wouldn't want to go that far.
Yeah, and I think that's
Thank
why
you.
it's going to be important, you know, when we formally release the volcano plot, is to make sure that it's accompanied by some instructions, you know, with those caveats. So that people don't make the mistake of, you know, only looking at the events above the threshold line, or
Fair enough.
you know, assuming that it's magically has some statistical meaning.
This has been Ben Duncan from AbbVie. And that this topic, of course, is very difficult, and there's a lot of controversy and all sorts of things around it. You know, I still think it's a useful tool to have in the right context, but maybe in the final version, maybe there's some optionality around having the users specify whatever level that they want to put on the line, on the plot, I should say. You know, and the same thing with, like, when you're talking Bayesian statistics, where you have posterior probabilities, you have different threshold levels. I think they can be useful, but it's just, you know, we just have to, like you said, provide useful instruction as to what these numbers mean. But I would hesitate to just completely abandon it, because I do think it's useful to have some sort of discussion around thresholds.
Yep.
Thank you, Ben. Thank you, Mary. And now we don't have so many minutes left. I'd like to turn it back over to Michelle.
Okay, yeah. So as we bring the first quarterly scientific webinar of 2024 to a close, so first let me express our gratitude to the presenters and the discussant, including Jane, Spencer, Natalia, Princeton, and Cecilia. So the ISG, alongside with other task force within our safety working group, continuously innovate and develop new methods to enhance safety evaluation throughout the drug development life cycle. So our working group is sponsored by ASA Biopharmaceutical section. If you have any questions about our working group, please feel free to visit our website or reach out to us directly. So thank you for your active participation, and stay tuned for details about our next webinar. Okay, I will close our webinar here.